Hi, everyone, and welcome to track 2 of the symposium, which focuses on frontiers in clinical functional brain imaging. My name is Jordan Harrod, and I'll be your moderator for this session. The session will be a Zoom webinar, so as an attendee, you will be able to see and hear presenters and panelists, as well as their presentation materials, but you will not be seen on camera unless allowed by the session host. You will, however, be able to interact using the Q&A, the chat, and the hand raise functions at the bottom of your Zoom window to address your questions or comments to all attendees or directly to the speakers. We'll begin with talks from our 4 amazing speakers, followed by a moderated live Q&A. So as the speakers give their talks, feel free to ask questions using the Q&A feature at the bottom of the window, and we will address them during the moderated Q&A session at the end. For our first speaker, we have Dr. Haoqi Shan, an instructor in the neurology department at Massachusetts General Hospital and Harvard Medical School, who works in the Computation Clinical Neurophysiology Lab and Clinical Data Animation Center, led by Dr. M. Brandon Westover. His work focuses on developing a sleep EEG-based brain index for tracking the deviation from normal brain aging and its analytical validation in cognition, mortality, neurodegeneration, and neuroinfectious diseases. Thank you, Dr. Shan.
Thanks, Jordan, for the great introduction. So where's my slides? Yeah, cool, thanks. So basically, as you have heard from the keynote session, that Dr. Haoqi already mentioned a lot about brain age. So his approach is based on MRI, and here I will present another perspective from predicting brain age from sleep EEG for tracking brain health. So first of all, why sleep? Well, the brief answer is that sleep changes with age. So as you know, many people, if you have kids in your home, or if you still remember when you were a kid, so the pattern of sleep for a newborn baby is quite different. There's no obvious circadian. And then as you grow older and even older, the bedding time will be earlier, and then the waking up time will also be earlier. So that, elder people tend to have shorter total sleep time. So basically, sleep is recorded with multiple electrodes, including the one that is recording brain activity. So we call it sleep EEG, electroencephalography, sleep EEG. So here is an example of the spectrogram of sleep EEG, where on the x-axis, it's time in hours, the time you spend from the onset of sleep. And then we have this y-axis, which is frequency. And this frequency can be divided into several bands, including the slow delta band up to spindle band. Actually, there are even more bands on the top. So each band represents a different meaning, and it's related to different functions of the brain. And also you can see here on the top half of the figure is that we have 5 sleep stages, and W means awake, and then R means REM, which stands for rapid eye movement, that is when you have the most vivid dreams happening. And then N1 up to N3, it's so N1 is the light sleep or drowsiness, and then N3 is deep sleep. It's a progressive deepening of the sleep. So as we know that people spend roughly a third of their lifetime in sleep, and is it possible that can we measure brain age or brain health during sleep? So if we could do that, we have several patient benefits. First of all, the brain age, of course, we need to validate it, but if we can use it to track brain health, and it has to be regularly and very easily, so that people can track their brain health, for different nights. And then if we were able to do this, we can, after some treatment or after some change in your lifestyle, or if you take some medications, how does this change affect the brain health? That's also very helpful for patients. And then for the research side, we have several research questions. For example, if you could measure brain age from sleep, what is the normal range in healthy people? So the range in terms of night-to-night variability, and also in terms of compared to other people. Um, and also if you have several disease, how abnormal is the brain age? For example, there could be some diseases, as mentioned earlier by Dr. Enkenhoff, that, for example, Alzheimer, that brings the brain age up to 8 years older. And then there could be other diseases which, like you can measure the progressive change in its effect on brain age or brain health. Um, and then how is this related, for example, to clinical outcomes, including risk of, for example, dementia or even mortality? And the last and the most important question is, what is the biological basis of this measure? So is it measuring, for example, genes or different other kinds of phenotypes? That is the remaining question. So here we can see there's a just as a more further step into the sleep to introduce sleep further. So here we have here two columns. On the left, it's male. On the right, it's from female. And these are the spectrograms that you saw on the first slide. And then we have 5 sleep stages, each one on each row. And then the x-axis, now it's age. So as you can see, there is an age-dependent change in the spectrogram. So as we said earlier, that we already know that sleep pattern changes age, including this, bedding time and wake up time. And then, so just to illustrate the idea here is that, for example, this alpha peak frequency, it's around 10 hertz when it's 30 or 25 years old. And if people grow older up to 80 years old, that peak frequency will drop to 8 hertz. Although that looks like a small drop, but it's quite significant and consistent. And then we have other features that change with age. If you measure the total power here from 4 to 8 hertz, that will increase with age. And then there's other bands, for example, the spindle band, and also the spindle density during this particular N2 sleep stage will also decrease with age. And then here, the delta band power is also decreasing with age. So there are quite many interesting changes that is age dependent. So the concept of brain age is that how can we utilize these changes, these age-dependent changes, summarize them into a number that indicates how normal or how abnormal you are. So basically, here we show two people, two subjects. So their age is similar. One is 44, the other is 48. And then as you can see here, around 10 to 15 hertz, there's a lot of light strips, light highlight parts here. So these fall into the spindle band, which indicates this person during their sleep, there's a relatively higher spindle amount or power compared to the bottom one, where you don't see as obvious as the top one. So we would say, because this subject has more spindle power, so spindle is related to memory consolidation, so that we would guess that his or her brain age is younger, it's a healthy brain, it's a good sleep. So the brain age is younger, and if you take the difference, if you take the brain age minus chronological age, it's negative number. And then similarly for the other patient, we would guess that the brain age is older compared to the chronological age, so that the difference, which we call it as BAI, brain age index, it will be a positive number. So just a few more details about how we develop this brain age model. So basically, we took brain healthy subjects from our Massachusetts General Hospital sleep lab. There's around 2,000 subjects. And then we fit a model based on that. So the inclusion criteria is that right now we are only focusing on adults, and it has to be a diagnostic sleep recording. And very importantly, we have to exclude brain unhealthy, patients from this group, so that our model is trained on the healthy cohort. So we exclude them, and then we fit a model which is basically a linear combination of different sleep EEG features, and followed by some function to avoid negative predictions. Um, and I think I forgot to mention that after we train this model, we use the Sleep Heart Health Sleep Study (SHHS) dataset for validation. So the nice thing about that dataset is that every subject has two sleep recordings around 5 years apart. So then, as you can see here, that we are doing the validation, because once you propose this biomarker, I think it's super important to be able to validate it rigorously in various ways. The first one is a cross-sectional validation, where we just compare the chronological age versus the predicted brain age. So basically, as you can see, the the mean absolute deviation, it's around 7.8 years. I remember from the keynote presentation from Dr. Eichhoff, where using MRI, it's around 4 years, and now it's around 7.8 years. So it's a bit larger, which I think it's reasonable due to the high variability of sleep EEG, which measures the function of the brain. Um, and then we need to do this longitudinal validation, as we mentioned that there is this sleep heart health data set where each patient, there's two studies apart. So then because it's 5 years apart, we would expect that their brain age is also roughly 5 years apart. And the other very important aspect is that we need to measure night-to-night variability. So this is a unique aspect in terms of sleep-based measures. As we just mentioned that in Dr. Eichhoff's presentation, where if you predict brain age using MRI, so then the MAD, mean absolute deviation, is around 4 years. And then in our case, it's larger. So we want to see, suppose you have multiple nights, how does that decrease? How does the night-to-night variability decrease? Yeah, so to do this, just to mention, we used 86 patients from a EMU unit, and then for them, we have multiple consecutive nights. Yeah. So we then go ahead and validate BAI versus dementia, because we know that dementia patients, they have advanced brain age. So here we have these many 5 groups of people. We have some definition to stratify each of the patient into five classes. and then we look at the average BAI for each class. And as you can see, as the different stages of dementia progresses, the mean BAI is increasing. Um, and as you can see here on the B panel, on the right-hand side, the people with dementia, if you take the average of their difference of in BA, the brain age, minus chronological age, that's also above the perfect line. And then we went ahead by further validating if we to see the correlation of BAI versus dementia after adjusting for other variables, and it's indeed still significant. And the other aspect is to validate BAI with MOCA, which is a cognitive test. So it provides a cognitive score from 0 to 30. And as you can see, there is indeed an active and significant correlation. But as pointed by Dr. Einkhoff, that yes, in our case, the result is also quite, the effect size is not that large, indicating there's a fair amount of individual variability in this measure. And the other aspect is that we used a different cognitive test, which is called NIH Toolbox, which measures different aspects of cognition. And it's roughly categorized into fluid intelligence and crystallized intelligence. So the ones that which I indicate by C is crystallized and F is fluid. So fluid basically measures the ability to learn new things, and crystallized intelligence means the ability to use the already learned, the familiar things. And then just as a summary because at the beginning we proposed several patient benefits and research questions. So the answer to some of these questions is that we have validated BAI on the for different aspects of brain health, and very importantly, we are going to validate it in home sleep devices so that we can track brain age easier. And yet the more interesting thing is that we have several other projects that involves different treatments and see how does BAI change. And in terms of research questions, what is the variability of the brain age? What is the normal range? So we have provided some numbers, including, in terms of longitudinal or cross-sectional variability. But the very important question, like within this number, how much come from biology? How much come from the noise in the recording? That has to be further studied. And we also showed that for dementia, if you still remember the box plot, the bar plot, there's about 4 years increase. And actually, in our other studies, we have validated other diseases. And how is brain age associated with clinical outcomes? I think so this is also a part of another this study which evaluates the association of BAI versus mortality, which you can refer to this paper. And then what does sleep-based BAI measure? So that's a really interesting question. And to really answer this, there is an ongoing effort to relate BAI to genes and also to some of the MRI-based brain age projects. And eventually, we would like to see like what's the biological reason of people who got higher BAI. Yeah, so thanks for all the PIs and our colleagues. It's a really fun, really interesting study that I hope can help, not maybe not close the gap, but reduce the gap of research and patients. Thanks.
Thank you so much for that great talk, Dr. Sun. And as a reminder, if you have questions for Dr. Sun or any of our other speakers, you can leave them in the Q&A and we will get to them after all the talks. For our second speaker, we have Dr. Susan Whitfield-Gabrieli, Professor of Psychology at Northeastern University and Director of the Northeastern University Biomedical Imaging Center. Her lab employs multimodal neuroimaging techniques to investigate the pathophysiology of psychiatric disorders with the ultimate goal of discovering biomarkers for improved diagnostics, early detection and prediction of therapeutic response, as well as the development of novel therapeutic techniques with the hope of augmenting currently available treatments. Thank you, Dr. Whitfield-Gabrieli.
Thanks so much, Jordan, for that kind introduction. It's a great pleasure to be here in this wonderful symposium. Today, we'll be talking about clinical translation of neuroimaging, specifically in how it might be related to early identification, neuroprediction, and novel therapy. So even before COVID, I believe we've been facing an epidemic of mental health in adolescents with psychiatric illness. We might know this from our own family and friends or our own clinic or reading the popular press, but it's truly been abysmal where we have so much teenage anxiety and depression these days, and we need to find ways to address it. So we know that there's many different challenges in diagnosis and treatment of mental disorders. I'm highlighting 3 here in red, which I hope that we'll be able to address at some point with neuroimaging. 1, detection is often late. It's often done by crisis. 2, and importantly, treatments are often based on trial and error, despite the fact that we know that there's great patient heterogeneity in treatment response. And thirdly, treatments often, like pharmacological treatments, often have negative side effects, and they may be only partially effective and therefore have high attrition. So there's many different ways to look at brain structure and brain function, as we know, but today I'll be highlighting connectomics, or how the intrinsic structural or functional architecture of the human brain is elucidated by white matter structure like DTI or resting state functional connectivity. Specifically, how can we use the intrinsic structural and functional architecture in ways in which we can use it for early detection? How that could promote early intervention and possibly even prevention of disorders. How can we use the intrinsic connectivity for neuroprediction that would lead us towards precision psychiatry? And how can we maybe use these novel, this brain imaging for novel interventions like real-time fMRI neurofeedback? So this is an iconic figure from Mike Fox and colleagues in 2005, where he used resting state functional connectivity to elucidate the default mode network. And we'll see here in the MPFC and PCC, the 2 core medial hubs of the default mode network are known to be related to self-referential processing and mental time travel. So these brain regions are more engaged when we're perhaps reminiscing about the past or thinking or planning for the future. However, in depression, reminiscing about the past may transform into rumination about the past, and thinking or planning about the future may transform into obsessive worry about the future. And in fact, we have shown years ago that the degree to which the DMN is hyperactive or hypercorrelated does correlate with various psychopathologies. We first showed this in schizophrenia, but now we and others have shown that the degree or magnitude of, say, the resting state functional connectivity, the self-reference nodes of the default mode network are correlated with psychopathology, like the transdiagnostic symptom of rumination. Mike Fox also showed that there's regions of the brain that are intrinsically anti-correlated, like the dorsolateral prefrontal cortex here in blue. And it turns out that the magnitude of these anti-correlations with the MPFC and BLPFC vary across time and vary across people. And they give great insights into brain health and illness. For instance, we have shown that the degree of anti-correlations correlate with better executive function. We know that the anti-correlations develop selectively over development as does executive function, and unfortunately decreases with aging. But we also know that there's a significant reduction in these anti-correlations in many psychiatric populations that have cognitive impairment. So you see a kind of a functional dissociation, if you will. You have the hyperconnectivity, the self-reference nodes correlate to psychopathology, whereas a reduction of anti-correlations correlate with cognitive impairment in many psychiatric disorders. So how can we use this in terms of clinical translation? Well, we know that many current treatments for disorders are inherently reactive to suffering. wouldn't it be great if we could use neuroimaging to have a proactive approach and identify brain network pathologies in children who may be at risk for developing a disorder? So Mike Grecius and colleagues have shown that adults with major depression exhibit a hyperconnectivity of the default mode network in the subgenual anterior cingulate, which we know is related to depression. And there's more glial cells, and this cross literature showing that this is a really key region in depression. And the degree to which the DMN ropes in the subgenual anterior cingulate correlates with the duration of the current episode of major depression. One might ask then, is that pathology reflecting the current state of depression, or is it reflecting a neurobiological trait? So we set out to answer this question, and we looked at children who were at risk for major depressive disorder. And at risk meaning at familiar risk, so they had a parent with major depression, 8 to 12-year-old children. We placed the seeds in the self-reference nodes, so we had the MPFC and PCC seed. And what we found was that children who were at risk but had no mental diagnosis, the children who were at risk for depression had hyperconnectivity of the self-reference nodes in the DMN to the subgenual. So they are reflecting a very similar pathology as adults who are already diagnosed with MDD. Moreover, the degree to which the self-reference nodes of the DMN, the MPFC and PCC are correlated at rest with the subgenual ACC directly relates to internalization, which captures things like anxiety and depression. So we wanted to extend this idea a little bit further and ask the question, could we use neuroimaging to identify biomarkers early in life, even in children who may not be identified to be at familial or phenotypic risk, that might predict their subsequent behavioral trajectory? So as I mentioned, we know that the DMN anticorrelations grow selectively over time during development, and we know that they're related to executive function. So we asked the question, in young kids at 7 years of age, 94 kids, would the baseline connectivity, even though on average they're positive, the baseline connectivity that MPFC and the DLPFC predict changes in attentional problems. And that's exactly what we found. So here's a scatter plot. Each red dot is a person. The degree to which the MPFC and DLPFC is negatively correlated or positively correlated at baseline predicts changes of attentional problems. So those individuals who had more negative correlations at baseline got better attention, those individuals who had more positive correlations at baseline got worse, and many didn't change because it's a normative pediatric sample. We also found that the region of the brain that we know is related to depression and bipolar is the subgenual. We found that weaker subgenual resting state connectivity to the DLPFC predicted progression of internalization, again, which captures symptoms like anxiety and depression over 4 years in this normative pediatric sample. In other words, the weaker is connectivity at 7 years of age predicted worsening of anxiety and depression 4 years later. And in neuroimaging, we always want to try to replicate and or extend our findings. And we did replicate this finding when we went back and looked at the children who were at risk for depression. We found again that the degree of subgenual ACC and DLPFC connectivity predicted worsening of anxiety and depression 3 years later. And these resting state metrics actually predicted who was going to convert to depression. Secondly, teens with anxiety and depression also had this phenomenon where the baseline, the subgenual DLPFC correlation predicted worsening of anxiety and depression 1 year later. And so this finding that the subgenual ACC DLPFC connectivity predicts worsening of anxiety and depression fits well with the current literature, such as Mike Fox's research team, who has shown that the region of the DLPFC that's maximally anti-correlated to the subgenual ACC has the best target for treatment of transmagnetic stimulation, to get better treatment effects there. We also know from Helen Mayberg that she can have better treatment effects for major depression by doing deep brain stimulation at the intersection of 4 relevant white matter tracks. And so we capitalized on that finding, and we looked at the white matter integrity as measured by FA and tried to add that on to the resting state predictions to be able to predict the future trajectory of anxiety and depression. And we found that there was a significant improvement when we did a multimodal prediction model. And that brings us to the next topic of precision psychiatry and multimodal prediction. So here we have a different sample, this time of individuals who have social anxiety disorder. We have baseline fMRI task activation, diffusion weighted imaging, and resting state functional connectivity. With 39 patients, they all had cognitive behavioral training, and we had our initial behavioral assessment of anxiety was the LSAS. And what we found was the LSAS actually already predicted the treatment response significantly. So it already accounted for 12% of the variance of treatment response. So one question is, does neuroimaging even add anything at all? What we found was, well, yes, in the case it did. We found a cluster here in the occipital temporal region where the fMRI activation in that region predicted treatment response. And when you added that activation to the initial severity of anxiety, we then accounted for 41% of the variance of treatment response. So we had a significant improvement of treatment response. Now we're looking at the same subjects, but now rather than looking at fMRI task activation, we're looking at intrinsic connectivities measured by resting state and diffusion weighted imaging. So here we did a hypothesis-driven bilateral amygdala resting state functional connectivity because we know it's the brain locus of dysfunction of social anxiety. But we know from the previous study that the brain regions that may be associated with pathophysiology of a disorder may not be the same ones that predict treatment outcome. Like it wasn't the amygdala last time that predicted treatment outcome. So we also did a data-driven agnostic prediction model, and we also used the white matter integrity as measured by FA and PCA of all the diffusion weighted imaging measures for the inferior longitudinal fasciculus. And what we found is that the brain connectivity, so the intrinsic resting and structural functional connectivity, significantly improved the prediction of treatment response. So let's shift gears a little bit. And now that we know these resting state networks are plastic, how can we modulate them perhaps with other behavioral modifications? We know that mindfulness meditation significantly reduces the default mode network because it really it helps us focus on the present as opposed to be hashing the past or worrying about the future. So we set out to do an 8-week mindfulness meditation intervention with 6th graders in the Boston school districts. And what we found was that there was significant modulation of the brain networks, including increases in the DMN or default mode network anticorrelations. And importantly, what we also found was there was an significant increase in sustained attention as measured by the START task, as well as decrease in self-perceived stress and the degree to which the brain networks were modulated correlated with degrees in behavioral modification. So it's really nice that we have a behavioral modification with the mindfulness meditation, but you don't have any visual feedback as to how you're performing when you're doing mindfulness meditation. So we are now trying a new technique. Well, it's not that new actually, but we're trying real-time fMRI neurofeedback to give people a visual understanding of how they're modulating their brain networks when they're performing mindfulness meditation. So 2 key questions: can people learn how to modulate these brain networks? And we and others have shown that's true. And the more important question is, does this learned regulation of activation or correlation between these regions actually affect the mental processes mediated by these regions? And we have shown that that is true as well. So the way the paradigm works is people come in for a resting state scan, which allows us to identify their personalized networks. And then we have the subjects lying in the scanner. While they're doing mindfulness meditation, the experimenter tracks the bold signal of the relevant resting state networks, like the default mode network and the frontal parietal control network. That signal is then transformed into a ball task, which the person is performing while they're doing mindfulness meditation. And the idea here is when the default mode network is pushed down by mindfulness meditation, then that means that the brain state is in a good state, and so the ball moves up. Whereas if the default mode network is higher than the frontal parietal control, then the ball moves down. That's in a bad brain state. So I'm just going to show you a movie of a patient who was performing this. And I just wanted to call your attention on the left, what you'll see with the experimenter seeing, tracking the bold signal from the default mode network, which you'll see in green, and tracking the frontal parietal control network with the central executive network in purple. And the ball moves up a hop size proportional to the magnitude of the difference in the bold signals. So the game goal is to move the ball up into this yellow circle when their brain is focused. And when that does happen, the circle will shrink to make the task dynamically more difficult. So here's an example of a patient who was performing this task. So you can see here, right now he starts mindfulness meditation. And you can see that, I think, he's pushing down DMN here in green because he's performing mindfulness meditation. He's pushing the ball up, it goes into the yellow circle, and the circle shrinks to make the task dynamically more difficult. So you can see you really successfully push down the DMN. That has nice behavioral consequences and brain modulation consequences. So when we looked at the pre-post connectivity, what we found was that there was mitigation or a decrease in the hyperconnectivity, the MPFC and PCC, that was decreased here. And we found an increase in anticorrelations between MPFC and DLPFC here, and MPFC and dorsal ACC. So now we kept capturing both psychopathology and cognitive impairment. We did not find this network modulation, however, when we used the Shan feedback. And importantly, the degree to which the brain networks were modulated correlated with decrease in symptoms. So Aaron Kusi is leading our team in trying to identify electrophysiological signatures of these DM and anticorrelations so that we can scale this intervention up. And we're also trying to scale this intervention up by extending this into different patient populations, including teens with anxiety and depression, ADHD, and at risk for anxiety and depression. And we believe that this mindfulness meditation, real-time neurofeedback intervention may provide a non-invasive kind of circuit therapeutic that could be transformative for teens with anxiety and depression, and even for people who don't have mental disorders yet, but may be at risk for developing. And that might be an unprecedented opportunity for them to learn how to mitigate their own brain networks and symptoms. So in the future, could children get help before they're in crisis through early identification, get treated more effectively with better precision to available therapies, and possibly have options for even novel therapies? So I wanted to thank all of my colleagues who have made all of this work possible. Randy, thank you for having this symposium, and thanks to all my other colleagues at Northeastern and MIT and all the medical schools with whom I'm associated. Thank you.
Thank you, Dr. Whitfield-Gabrieli, for that great talk. And I can see people are already throwing questions down in the Q&A, so definitely continue to do that as we go through the rest of our talks. Next, we have Loraine Franke, a PhD student at the University of Massachusetts, Boston in the Machine Psychology Lab. Her work focuses on the visualization of diffusion MRI fiber data in an efficient and interactive way that allows for comparisons between different clusters or bundles and subjects. Thank you, Loraine.
Oh, sorry. Hi, everyone. Thank you for the introduction, Jordan. Welcome, everyone. So today I will present on a topic of interactive visualization of complex brain imaging data elements to gain individual patient-level insights. And in this context, I'd like to present a tool that we have developed. It's called FiberStars, and it's a tool to for the visual comparison of diffusion tractography data between multiple subjects. So a bit about the background. With tractography data, which we acquire from DMRI scans, which you can see in the animation on the right bottom, we want to directly compare regions of interest in the brain across multiple patients. So for example, if we try to compare a group of subjects suffering from a disease versus a group of healthy subjects. So our goal was to facilitate the analysis of this high-dimensional diffusion MRI data in a web-based tool where we pair 2D and 3D visualizations, especially in an interactive way for the user. So we also want that users do not necessarily require a detailed understanding of complex relationships and the patterns in the data, because sometimes you have a data set and you just want to explore and query it. So our challenges is at first that we had large amounts of data. One attendee also asked a question about the ABCD data set data. So we also tried to apply our tool on this one. So we have 800 files of white matter tracks per subject, which is also called a cluster. And those data sets can have hundreds of gigabytes in size. And another challenge is also the different file formats, which differ from institution to institution. And it's also really important to understand the data, especially the values, numbers, scalars, and other measurements that come with those DMRI scans. And another challenge we were facing is the question how many subjects can be visualized effectively. And such as what is important to visualize at all. So what do researchers and neuroscientists or doctors want to know? So we wanted to create a web-based tool because it has some advantages, especially in terms of data sharing and that no download or client-side installation is necessary, and also that it's accessible through different devices. And for the web-based tool, we use React.js, which is a state-of-the-art JavaScript framework for good usability, and it's really easy to add more components and more features. So for the 2D animations or 2D visualizations, we use D3.js, which is also a JavaScript library, and VTK.js is used for the 3D anatomy, which is displaying the fiber tracts, which you can see here. And it's basically measured with XYZ coordinates for each fiber. So one fiber is measured with XYZ coordinates. We use multidimensional scaling, or MDS, for creating scatter plots. And we also use compression algorithms. We recently presented TrackO, which compresses tractography data up to 28 times without a loss. So let's think of a concrete example. If we want to conduct a clinical study to analyze factors combining with a pathological finding, so we have DTI data sets of healthy subjects, which are the controls, and patients suffering from an auditory verbal hallucination, which is a symptom that can occur in patients with neuropsychiatric disorders or other diseases. And we have the hypothesis that fractional anisotropy, which is called FA, Susan talked about that before. It can also be measured, I think, people having depressions. So this is something we get with the DMRI scan, and it explains in general just the diffusion of water molecules in the brain tissue. It reflects also the fiber density, axonal diameter, and other measurements. Then we scan our patients, so the healthy ones and the ones diseased. And we receive the DMRI data with the fiber tracts of each subject's brain. And we want now use a tool to explore the data and test our hypotheses. So at first, we start using the tool of our data set and get into the projection view, which is an entry point for the user. At first, there's no in-depth knowledge not needed of the user. So the right image shows the projection view user interface, and you can see that one point is one fiber tract. So we have an algorithms, for example, pivot MDS, which create a scatter plot with each point being one cluster, one tract. So this is a really high dimensional space. So the user can select dimensions, and the projection view will group those clusters or points accordingly. So here, for example, we have selected 5 subjects from our data set. And let's assume that 4 of them are healthy and 1 of them has a disease. And we say that subject 101, so the light blue label, is the one suffering from the symptoms. So if we now hover over one of the points in the scatter plot, we receive more information on the white matter tract in a small window, and also some demographics of that patient. So in the projection view, it will also help us to examine and explore a larger number of fiber tracts. So on the left, we can see that we have chosen FA1 and FA2, which are some measurements of fractional anisotropy. And we have, again, selected the 5 subjects. So when the scatter plot is adjusted and the points are adjusted accordingly, we can explore how the point clouds are looking. So now we can see that we have found some outliers, which seem to be a bit more on the left than the other points. So with the cursor, we can perform rectangular brushing, choose those outliers, and then we get to the multi-cluster view. The multi-cluster view enables a comparison between multiple subjects and multiple fiber clusters in a 2D plot. So here's an example. There's only one fiber tract for each subject, which is highlighted in violet. It's the forceps major. But we can also display many other tracts as well. So for each cluster, one 2D radial plot is shown with a normalized scalars on each axis. So when comparing multiple radial plots, so outliers can easily be spotted, especially when we show multiple 2D plots next to each other, because radial plots are less complex visualization. So from our example, we had subject 101-006, which was the one diseased. And we can see that FA2 and FA1 are a bit more to the middle, which means that the values for these subjects are lower. So in the next step, we consult the 3D anatomy of the fiber clusters. So we can see that the tractography data of 5 different subjects with the 3D anatomy is shown. And we can see here in the middle, we have again the same subject, which was deceased, and our healthy controls next to the deceased one. At first, we check again the 2D plots. So in the bottom, you can see that we have again the one we saw before as well, that FA1 and FA2 seem to be lower. And we can also see that the center of the tract is compared to the other subjects not highlighted in warmer colors. So you can see that the 4 other ones have more warmer colors, which means higher values. So as we have chosen color by FA2, so we can color the whole fiber tract by a measurement that we are interested in. We can see that for this subject here, it seems to be a bit lower. So the values are more blue or greenish in the middle. So there might be something that is not normal in this one. And the next thing we could also think about is if we compare demographics, for example, we can see that this one has an age of 35. The other subjects seem to be a bit younger. Maybe this also has something to do with the lower FA values. So we could also test the new hypotheses. Are older patients or subjects, do they more suffer from those lower FA values? Yeah, and in summary, so the tool can help to test hypotheses, also to generate some hypotheses and answer research questions by comparing those fiber tracts and differences among multiple subjects. So we know that DMRI is a very specific use case. It can also be, maybe also be applied to other neuroimaging techniques and also to other fields with similar visualization challenges, especially when we deal with multidimensional data and 3D shapes. So this is also the case in biology or in astronomical data. Our tool is openly available, so it's on GitHub. And I've also put in the link here and the paper. And yeah, I'd like to thank Randy for letting me present on the symposium. And thank you to my co-authors and people who supported me. Thank you.
Thank you, Loraine. And as I mentioned earlier, if you have questions, feel free to drop them in the Q&A. I can see that there are already some down there. Finally, we have Dr. Shan Siddiqi. Dr. Siddiqi is an instructor of psychiatry at Harvard Medical School and director of psychiatric neuromodulation research at the new Center for Brain Circuit Therapeutics at Brigham and Women's Hospital. He completed his general psychiatry residency at Washington University in St. Louis, a neuropsychiatry fellowship at McLean and Massachusetts General Hospital, and a research fellowship at the Berenstain Allen Center for Non-Invasive Brain Stimulation. His research is focused on personalizing TMS targets using brain circuit mapping and clinical phenomenology. Thank you, Dr. Siddiqi.
Thanks for that introduction. So today I'm going to be talking about what we're doing to sort of develop circuit-targeted therapeutics using some of the approaches that you've heard about earlier. The basic idea is let's take a lot of this brain imaging stuff that everybody's been working on and developed to a great deal to make it quite useful and see if we can translate that into treatment targets. And what I'm going to be talking about a lot is something called TMS, or transcranial magnetic stimulation, which you may or may not have heard of. It's a tool that uses electromagnetic induction to focally induce action potentials with a radius of about 1 centimeter. The effects are believed to then propagate to distributed brain circuits. So it probably distributes throughout a big chunk of brain. And one of the questions that we have to answer is how do we map those circuits? So we commonly use single or paired pulse TMS in physiologic studies. For example, stimulate the motor cortex and observe a twitch in the thumb. But more commonly, we use repetitive TMS. When I talk about TMS, I'm going to be talking about repetitive TMS today. And that can induce long-lasting changes for hours or days. Our TMS of the dorsolateral prefrontal cortex, which is around here somewhere, is an effective treatment for depression. And in fact, our TMS is FDA approved, or different types of TMS are FDA approved now for major depression since 2008, for migraine with aura since 2013, for OCD since 2018, and for nicotine use disorder just since middle of last year. However, there are a few challenges, especially when it comes to targeting TMS. Right now, we literally target TMS using a tape measure with scalp landmarks. We make somebody's thumb twitch by stimulating their motor cortex. We pull out a tape measure, measure about 5 or 6 centimeters in front of that, and say that's probably about the right spot to stimulate. Now, we know that's not the right way to do it, but it works. It works clinically, so that's the best thing that we have. Now, these landmarks were originally designed based on the approximate location of brain lesions that cause depression. So that makes sense. People found that brain lesions that cause depression tend to be in the dorsolateral prefrontal cortex. So maybe if you stimulate that region, it might alleviate depression. But now, since then, the same landmarks have been used for all sorts of other things: PTSD, generalized anxiety disorder, addiction, even Parkinson's disease and chronic pain. And that's led to mixed results. And it doesn't necessarily make a lot of sense to be treating generalized anxiety or addiction based on the location of lesions that cause depression. We need to be able to find a better framework that's targeted to each specific disorder, or each specific syndrome, or each specific symptom. Now, we actually have the ability to target precise locations in any given patient's brain using this thing that we call stereotactic neuronavigation, where you can register the patient's brain MRI into a 3D camera system and figure out where the patient's brain is with respect to our TMS magnet. However, all the precision in the world is useless when we don't know the right target. We're still trying to figure that out. And the way that we approach that is by trying to find a reliable framework for translating lesions to TMS targets for different disorders beyond just depression. So we usually do this using data science, but now we're actually translating our models into clinical trials. So now this is a question for precision medicine. On Monday afternoons, I'm a clinician. I'm sitting there thinking about how can I best treat my patient. The rest of the time, I'm a researcher thinking about how can I use my favorite tools and optimize my favorite tools. And so I think this whole approach gives us an opportunity to sort of bridge that gap. And the way we start to do that is by using brain lesions, among other things. So traditionally, brain lesions have been used to study the causes of neuropsychiatric symptoms. And that's different from the correlates that we find when we put somebody in the scanner and observe what's different and what's the same. The reason is because, for example, in this instance, if you find 3 patients who have brain lesions in 3 different spots, and you find they all have the same symptom, what people say is if you look at where those lesions overlapped, that's probably the spot that causes that symptom. That's a reasonable inference in theory, and it works sometimes, but very often in practice, it looks more like this. You have different parts of the brain that can be lesioned and cause the same symptom. So for a long time, people assumed the lesion probably has nothing to do with that symptom because they're all over the place. However, we've found that sometimes when different lesions cause the same symptom, there can actually be something in common. Sometimes those lesions fall into the same network. Now, you've heard a little bit already about diffusion tractography, which we're showing on the left from Loraine, and you've heard from Sue about functional connectivity, which we're showing on the right. So I'm not going to recap all of that, but the basic idea is these lesions might be connected to each other via common fiber tracts. They might be connected to each other via common functional networks. And I'm going to talk more about functional networks today, just because that's what I spend more time thinking about. But what we're trying to do with this information now is figure out how we can get different types of causal information and integrate it into a common model. So I told you about brain lesions. You can look at the intersection of brain lesions to figure out what brain region causes something, but you can also look at whether they intersect with the same network. You can extend that approach to other things that are causal, such as transcranial magnetic stimulation. You stimulate a specific spot in the brain and you see an outcome. You stimulate a different spot in the brain and you don't see that outcome. It's reasonable to infer that that region was causally implicated in that outcome. We can do the same thing with deep brain stimulation, which I haven't talked much about yet, but it's the same principle, except it's more invasive, but also more precise and potentially more powerful. And at the end of the day, we can put all these into a common model to say, if lesions, TMS, and DBS are all modulated in the same circuit to modify the same symptom, that gives us a lot of a really strong causal inference about the involvement of that network in that symptom. So here's how we started doing that. We took 14 data sets with different kinds of causal sources of information, total of 713 patients all around the world, including 196 Vietnam veterans with penetrating brain lesions, a couple hundred stroke lesions all the way from St. Louis to Melbourne, 50 hemorrhagic stroke lesions from Chicago, a bunch of different TMS sites all over the dorsolateral prefrontal cortex. I told you earlier that we use a tape measure to target TMS. That's imprecise, but the benefit of that is that it introduces incidental variability in where patients are incidentally stimulated. So everybody's actually stimulated at a slightly different spot. If we localize that spot using a brain MRI, we can actually study the effect of that variance and figure out what happens if you stimulate the right spot versus the wrong spot. And so we took 4 different TMS data sets with 150 patients. Also, about 44 patients who received deep brain stimulation for major depression, which Sue mentioned earlier. People have been doing it at Emory, all the way up to Berlin. as well as 32 patients who got deep brain stimulation for Parkinson's disease, but also measured their depression outcomes, and 25 in the Netherlands who got deep brain stimulation for epilepsy, and also measured depression outcomes. And the question we had was, can we find a common network that mediates improvement in depression across all these heterogeneous data sets, brain injuries, strokes, TMS sites, DBS sites for Parkinson's, epilepsy, and major depression? And here's how we did that. We said, let's take a few brain lesions, for example. Here's 3 examples of brain lesions. Look to see what those lesions are supposed to be connected to using a normative connectivity diagram. So just without individualized connectivity data, in this analysis, we assume that people are more similar than they are different. And we've looked carefully at this and found that that's true. There are inter-individual differences, which we think a lot about, but the similarities are stronger than the differences. And so we find that we map the connectivity of each lesion. We look to see if the patient developed depression or not. And then we get a map of connections associated with severity of depression after a brain lesion. If your lesion is connected to this network, it affects your risk of depression. Similarly, we do the same thing with TMS. We map out a few different TMS sites, look to see what they're supposed to be connected to, see how much the patient improved in terms of depression. And we get a map of connections associated with antidepressant efficacy of TMS sites. TMS sites connected to this network are more likely to improve depression. Finally, we do the same thing with deep brain stimulation. We look at a few different stimulation sites. DBS sites are usually placed using in neurosurgical procedures, but they don't have very precise coordinates of exactly where they want to place it. They roughly place it in a certain brain region. So again, we have variability. Each DBS site has a slightly different connectivity profile. We look to see if the patient improved in terms of depression or not. And again, we get a map of the connectivity of DBS sites that led to improvement in depression. And sure enough, those maps look the same. The brain lesions are connected to brain lesions associated with depression are connected to the same circuit as TMS sites that improve depression and DBS sites that affect depression. And DBS and TMS are actually inverted, but for visualization, I put them on the same scale or the same, corrected the inversion here. And that's those similarities are statistically significant. The way we define significance here is we randomly reassign one patient's clinical outcomes to a different patient's neuroimaging. So we do a permutation test, and we reiterate it 10,000 times and show that the real similarity between these maps is much stronger than expected by chance. So what this tells us is there's a common circuit that's sort of causally implicated in depression. It's an effective therapeutic target, and it is actually a more effective therapeutic target than any of the targets that we've had so far. This paper is currently under review, but the next question we had is, if this works for depression, is this a framework that can translate further? So to summarize, we found that TMS, DBS, and brain lesions can all modulate a common brain circuit to affect depression. So thus, lesions can help us find new TMS or DBS targets. TMS targets can help us find DBS targets. DBS targets can help us find TMS targets, et cetera, potentially for multiple disorders. So we wanted to test that hypothesis. Is it really true for multiple disorders? We got some data for Parkinson's disease, specifically motor symptoms. We didn't have quite as much data here, but found 29 case reports of lesions that cause motor symptoms of Parkinsonism, 10 clinical trials of the motor cortex, of TMS of the motor cortex for Parkinson's, which were effective, and 2 data sets of deep brain stimulation for Parkinson's disease. And once again, they all seem to be modulating the same circuit to exert improvement in motor symptoms of Parkinson's disease. So the model goes beyond just depression. It seems like a model that'll help us figure out, identify new treatment targets for a variety of different disorders. So taking that further, we can really test anything. So we've used lesion network mapping for movement disorders such as hemichorea, psychiatric symptoms such as delusions of familiarity, other movement disorders like freezing of gait, and even more abstract phenomena such as criminality. We have a paper under review now about religiosity and spirituality even. So in theory, we could modulate a lot of those behaviors. Of course, that raises some ethical questions. But to take it a step further, we want to see if we can break down depression, because not all patients are the same, right? As a psychiatrist, I don't want to prescribe the exact same treatment to every single one of my patients. But there are different symptoms that different patients experience, and we wanted to see if we can map out different circuits that we can target for each one. So the way we did that was we took a few different symptoms. Here's some examples: decreased interest, feelings of failure, and insomnia. We did the same thing as we did before, took about 30 different stimulation sites in patients who had TMS for major depression. Look to see what those stimulation sites are supposed to be connected to using a normative connectivity diagram. And then look to see which connections were associated with improvement in each symptom. So if a TMS site was connected to this circuit, you had improvements in interest. connectivity to this circuit mediated improvements in feelings of failure, and connectivity to this circuit mediated improvements in insomnia. And it turns out we did this for 21 different symptoms of major depression, and overall they fell into two different clusters. So symptoms such as sadness, decreased interest, and suicidal thoughts fell into this first cluster, and changes in sleep, decreased interest in sex, and irritability fell into this other cluster. And you can see these maps kind of look the same. We combine these using a clustering algorithm into what we call our dysphoric cluster and our anxiosomatic cluster. And I'll preempt this question that we tried 5 or 6 different clustering algorithms, and they all give us the same answer. We tested this using permutation testing, rigorous statistical validation, which I'm happy to talk about if anybody wants to ask about it. But I think that's the boring part for most people. So then we were wondering, is this real or not? So we replicated it in an independent cohort of 81 patients. Again, low mood, decreased interest, and suicidality fell into the dysphoric cluster. Difficulty sleeping, decreased libido, and worry and irritability fell into an anxiosomatic cluster. So we combined these maps together and created what we called an overall targeting atlas. And here's what that targeting atlas looks like. It's the red spots, the orange spots are good for dysphoric symptoms like sadness and suicidality. The purple spots are good for anxiosomatic symptoms such as insomnia and decreased sexual interest. And in fact, this actually predicts prior clinical trial outcomes. So the more, we found 14 prior studies that had stimulated 9 different spots on this atlas. And the more anxiosomatic their stimulation site, the more anxiosomatic their clinical improvement. And the more dysphoric their stimulation site, the more dysphoric their clinical improvement. We also showed this in both of our data sets across individual subjects, not just across large-scale clinical trials. So next step, we're actually now doing our own clinical trial where we take patients with depression and anxiety, and we either stimulate them here at this dysphoric spot or here at this anxiosomatic spot. We just started this clinical trial. We've already enrolled 1 patient, and we have a bunch of people on the waiting list. It's been a wildly popular trial because we're offering a new treatment for depression and anxiety, which I probably don't need to tell this crowd is an important problem. So what are we going to do with this next? Future directions. First of all, I just told you clinical trial of dysphoric versus anxiosomatic targets. This was a future direction a week ago. Now it's sort of a present direction because we just started this trial. But if this is successful, it'll probably constitute the first conclusive demonstration that brain mapping can translate into real-world precision treatments. We can actually select the right treatment for the right patient based on a brain map. That clinical trial is underway. Hopefully we'll have some results to report in a year or so. We also want to expand our models to understand more behaviors and also incorporate the effect of inter-individual variability in brain topography. Individualized brain mapping is really fascinating, and it's a whole different can of worms, which I could talk about for hours, but I won't today because I only have 15 minutes. But if you're a student, if you're a postdoc, we've got a lot of opportunities for trainees. If you want to learn clinical TMS, learn about our clinical trials, do a longitudinal research rotation, or become an expert, we can help you learn any of those things. We're always looking for good people, postdocs, research assistants. If you're interested in collaborating, share your thoughts, share your data. I showed you data from over 1,000 patients today, I think, and only actually 0 of them were people who I actually saw myself. We rely very heavily on data sharing collaborations. And of course, if you're a clinician, you're welcome to come and learn more about TMS from our service. A lot of people helped us build what I showed you, and a lot of people helped pay for it also. If you have any questions, you're welcome to reach out to me via email. Twitter has become my primary mode of scientific communication these days. This is my website, and of course, chat box. Thank you.
Thank you, Dr. Siddiqi, for that great talk. So now we're going to move to the moderated Q&A session. And as a reminder for attendees, you can submit questions using the Q&A feature, and we will be pulling questions from the Q&A throughout the session. As a reminder to panelists, I'm sure that I can speak on behalf of myself and all of the attendees when I say that we're looking forward to your responses to these questions, but in the interest of trying to answer as many questions as possible, please try to keep your answers concise. And to give attendees time to submit questions, we're going to start with a discussion question for the entire group. So we heard in the keynote from Dr. Eckhoff, as well as in some of your presentations actually, about the importance of looking to work that others have done to inform our own. Having heard the talks from your fellow panelists, do you see any opportunities where your work would benefit from integration with that of another panelist? And we can start with Haoqi.
Definitely. I mean, because in my presentation in our work, we mentioned that this BAI can be an indicator for, after some treatment. So, for example, I guess for Dr. Shan Siddiqi's work, that could be an interesting point. For example, if we could see, like, if you have some treatment for depression, how does that improve sleep? And then in turn, how does that improve brain age and brain health? That could be a super interesting thing. And I think actually in our lab, we have also some projects investigating, different exposure and different treatments that changes brain health. So I'm sure that could be a super interesting thing to look at. Thanks.
Dr. Siddiqi?
I agree completely. I was actually going to say one of the same thing when you asked me that question, among other things, that it'd be a really interesting biomarker. And then I think we all benefit heavily from collaboration and talking to each other. I think Sue probably knows or has probably guessed that I already use her toolbox all the time. But she didn't talk about that today as much. But I'm really interested in seeing how a lot of our circuit targeted therapeutics will affect a patient's ability to engage a network on neurofeedback also. That would be a really cool thing to look. If I stimulate the default mode network, does it get it easier for a patient to engage that network, for example? And then hearing about Lorraine's cool approaches to thinking about how to parse out diffusion tracts. That's really important for making things scalable. We need to take similar approaches to making our functional connectivity tools scalable, so real world clinicians can use them. So yes, all of the above.
That sounds like there's a lot of great opportunities for collaborations here. Sue? So you're muted.
I see many opportunities for collaborations, and Shan and I have talked about, he brought up a lot already. I really like the idea of whether TMS can increase the response to, say, behavioral modifications like real-time fMRI neurofeedback. Also, I think Haoqi's presentation on sleep is hugely important, also in how it relates to depression, not only for baseline biomarkers and for treatment effects, but how things like TMS, DBS, real-time fMRI neurofeedback, how those specifically modulate sleep patterns, especially in the default mode network, because we know the MPFC PCC connectivity is decoupled during slow wave sleep, and that decoupling, I think, might be modulated in different ways in different psychopathologies. So I think there is tremendous opportunity to collaborate with Haoqi and looking at those sleep dynamics and different sleep stages. And Lorraine's work is fantastic. And as you know, we like a lot of multimodal prediction. So her diffusion work is extremely important as well.
So, and last but not least, Loraine.
Yeah, thank you. I think so too, Susan. So yeah, especially also when we look into the depression population, for example, we could use the data sets, for example, Sue had, look also if there are differences in certain fiber tracts, or what Shan said as well, looking into the scalability to visualize the whole thing. So I hope that with those tools and stuff, we can support each other.
Great, and to get to some speaker-specific questions. So for Haoqi, there were a few questions about generalizing your model, specifically for populations with disease and across effects of different drugs, for example, and comparing it to MRI-based measures of brain age, like those mentioned in your talk and in the keynote. So can you talk a little bit more about that?
Sure. Yeah, those are super important questions. I mean, for example, for patients who are taking drugs, or a patient with different diseases, I think, well, you have to think about this in two ways, in terms of model development and model validation. So I guess, of course, in our model development, we don't want to have those patients, because otherwise their EEG, their sleep EEG is changed, is modified by these drugs or diseases. So that the model will learn to compensate, to try to make the unhealthy people to look to predict a normal age. So in terms of model development, definitely we want to keep the cohort as healthy as possible. And then for validating the model, I think it's, once the model knows what is normal, what is abnormal. Then when you apply to people taking drugs or people with diseases, and then you will see a deviation. You will see their brain is having different sleep patterns compared to another person without that disease or not taking that medication, but at the same age. So that could be really beneficial. Yeah, but just to coming back to make sure the model development cohort is really healthy, but then how healthy is healthy? That's also a difficult thing to define. So that's why right now we are only constraining to people who are brain healthy, who don't have significant neurological or psychiatric disease. But it's for sure there's many other type of disease that exist in our development cohort, which we haven't really controlled for. Just a
Thank
very
you.
brief. Oh yeah.
Oh, sorry.
Oh no, just responding the MRI question. Yeah, I think future direction will be relating BAI to, sleep-based BAI to MRI-based BAI to understand, for example, the stationary part of the variance. So how much is due to the structural changes? How much is due to the night-to-night variability changes? So that could be very important. Yeah.
Thank you. And we have a question for Sue. So some results from the ABCD trial suggest that given natural variation, the sample sizes needed to draw robust inference on most imaging-based measures are in the many thousands. How do these results affect how you think of these kinds of studies that you've shown here, where the N is on the order of less than 100? And in which settings would we need thousands of subjects, and which questions can we answer with 50 to 100?
That's a very packed set of questions, which are all excellent. Yeah, there are a number of papers that illustrate that you may need thousands of people to be able to get reliable brain behavior relationships. Is an excellent point. And I think what we need to do actually is to shift gears and pivot, do a little bit of a paradigm shift, and then go into a deeper phenotyping on the individual level. So get multiple samples on the individual treatment, diagnosis and treatment based on the individual. And that can be done with experiential sampling. And ultimately, I think that's kind of a direction that we need to go. I do think you can get somewhat reliable correlations that have been, that's why I really spend a lot of time replicating it in many different samples, like the one I showed you. There was replicated in many times and really does predict clinical outcome, which is ultimately what we want to do. But I do think that what we need to do as a field, or at least what I think I would like to try to promote, is a lot deeper phenotyping on the individual basis, and do actually the less large studies with thousands and thousands and more deep studies within the individual.
Great, thank you. And to jump back to the whole panel for a question, based on the projects that you each presented on, what do you think is the most exciting future opportunity in neuroimaging for assessing brain health, and what's the biggest challenge or limitation that comes with it? So we can start with Loraine.
Yeah, so especially I think that we have really large data sets, not only what I presented in DMRI in general, also maybe fMRI, other neuroimaging techniques, which is a big challenge. And yeah, of course, we have improving techniques and computational power, but however, there's still, room to improve. And the other thing is also, which I mentioned, that we have different file formats, and those file formats are different from institution to institution. And so I don't know if we have to come up with like an overall file format, which could be helpful. Or, I think that's still a challenge. And for neuroimaging in general, I think that it's really interesting to see the different disciplines, like how can we combine stuff, and how can we, find like findings from other contexts, how can we apply it to our field of research. That's pretty cool.
Great, how about you, Shan?
So there's a lot of exciting opportunities potentially. I think right now the biggest thing that's stopping us from moving forward is having the people to do it. So the thing that stops us, it's we have the challenge that we want to overcome is figuring out how to actually translate some of these these ideas to real treatments. And we have way more ideas than we have hands and brains. So I think the challenge is recruiting great people.
And Yusuf?
I agree with both Loraine and Shan on this. Again, I think that there's so much individual variability, and that's quite meaningful, that getting a better understanding within each person's brain structure and function and physiology, and how that relates to the clinical symptoms to coalesce into a deeper phenotyping could be helpful in terms of future prediction for individuals.
Thanks. And last but not least, Haoqi.
Oh yeah, yeah. I think since all of us are proposing some sort of biomarker, I think it's given our ability, in this big data and advanced analysis, that really provides us, a number to describe some high-level thing that summarizes a lot of stuff that previously you couldn't measure. Just for example, if I take my example, previously a sleep doctor see a sleep EEG and immediately they can tell, this is good sleep or bad sleep. But this brain age index helps to quantify this concept so that you can easily and repeatably extract this information to summarize these high-level concepts into concrete numbers. And then there may be some more downstream algorithms that make use of these biomarkers to further, for example, do either control or other stuff. Yeah.
Great, thank you guys. We have a question for Shan. So what are your thoughts on the role of individual mapping and the future of TMS treatments for neuropsychiatric diseases?
Yeah, so those are, I'll answer each one at a time. Regarding the role of individualized brain mapping, so that's actually what I got started looking at, is individualized brain mapping. I thought it was the coolest thing in the world, that we could map the rest of state network boundaries at the individual level, and they're reliable enough, so you scan the same patient twice, you get the same boundaries. And it's exciting, but the challenge is that no matter how much precision you have in mapping the brain network boundaries, you still have to know which network you want to stimulate for any given patient based on their symptoms or clinical profiles. So we're incorporating both. We're trying to figure out how to relate individualized network topography and symptom outcomes. So I spent a lot of time trying to find the optimal brain network mapping algorithm and thinking about the right way to compare that to TMS targets. And we haven't yet gotten to the point where we're satisfied with saying that this clearly predicts clinical outcomes, but we do have a couple of studies ongoing on this topic. I've done a pilot trial using individualized mapping approaches in TBI-associated depression, and now we're replicating that in multi-center clinical trial to see if we can at least get it to work, and then we'll figure out why it's working. So in short, we're using it. We're trying to see if we can get it to work, and we think that the individualization is the reason why it's working, but we will tease that out once we have the data. Regarding the second question, the future of TMS for neuropsychiatric disease. So TMS is already widely used for major depression, and I see the question in the text box is asking if it'll become more of a standard clinical tool or if its main application will remain in research. So in fact, I would probably argue that its main application is in the clinic right now. I think TMS is much more widely used as a clinical treatment for major depression than it is as a research tool. The TMS device companies that are selling to clinicians are worth hundreds of millions of dollars now, and the TMS devices that are companies that are selling to researchers are worth $9 or $10 million. So I think it's already gotten to the point where it's a fairly mainstream clinical tool for major depression. As for using it for other things, which I also see in the question box here, such as essential tremor and Parkinson's disease, we've got the evidence. The reason why TMS isn't used for Parkinson's disease, despite strong evidence, is because it's just not anywhere near as effective as deep brain stimulation and or as durable. So it becomes a question of clinical equipoise at that point, is that it'll become used more regularly for other clinical disorders as we find that it's effective for different things. So as I mentioned, it's now approved for nicotine use disorder and OCD, and people are using it more and more for that. As we discover more and more indications, it'll be used more and more. So I do think that it'll become an increasingly standard clinical tool. What I usually say is that TMS is like psychiatric laparoscopy. You can use it to modulate any part of the brain. We just have to figure out what part of the brain you want to modulate for any given thing.
Awesome. Sounds exciting. So one question that I want to get to, since we have time for a couple more questions, I'd imagine a lot of the trainees here might be curious about this. What kinds of backgrounds and skill sets would be most useful for your work, especially to overcome current limitations? So why don't we start with Sid?
That's a great question. So it takes a village. So, right, Shan, for instance, a psychiatrist, you have the clinical aspect, you have the imaging aspect. So that imaging can range from physics to statistical analyses of neuroimaging data. And then there's the clinical aspect. So I think getting well-versed in what you're interested in well and deeply is a good approach. So basically, I think that the neuroimaging side of things from my perspective, understanding I don't think you need to understand the depths of the physics of the acquisition precisely, but the consequences of the acquisition parameters perhaps, and then how to do data analysis and what the implications are for those data analyses in terms of the clinical context.
Great. And you, Loraine?
So I think the same as Sue, especially like how the acquisition is made. Also, what I talked about DMRI, at the beginning, I was trying to understand how is the scanner working, like how do they get those data and what is important. So what do neuroscientists want to know? So yeah, and then also like building the bridge to what is important to visualize, and yeah.
Great. And you, Haoqi?
Yeah, so I think these days, including all our speakers, I think it's really becoming interdisciplinary, so that you either as a doctor or as a researcher, you have to understand both the clinical part and also the technical part, and also the scientific part. So I think it's really helpful if you want to become engaged in this field, then you should have close collaboration, close talks with people from these three fields. Because my background was originally in more machine learning part. So when I first come to the hospital, I see, oh, it's just data, doesn't mean too much. But once I start talking to doctors and even patients, I start to realize how important it is. It's really a good lesson for me and for many of you to learn. Yeah.
Thank you. And last, Shan, who had a great last slide, also hitting on a lot of these points.
Yeah, we have just within our lab, our lab group, we have psychiatrists, neurologists, psychologists, biomedical engineers, physicists, computer scientists, all sitting in a room talking about the brain, and it's a lot of fun. We would benefit from more different approaches. Like if there was a philosopher who wanted to come in, or a physicist, they could both equally find a home there. So what I often say is that neuroscience is the last refuge of the polymath. There's so much left to learn about the brain that every new approach is welcome. And recently I tried to sit in on a class about neuroscience and philosophy. And I realized that I wasn't following their discussion at all. Like clearly there's a whole different approach to it that I'm not even privy to. And so I think there's I can't even imagine the different kinds of backgrounds that can potentially contribute to the way that we think about the brain and the mind. And so I get excited if we have a job applicant, for example, who has a different background, who might have a different way of thinking about things, like, I don't know, political science or
That'd
theology.
be a really fun one. Yeah.
Yeah.
Awesome.
Yeah.
So we're gonna. Oh, sorry.
So I was going to say, one of my colleagues in our group, Michael Ferguson, is getting ready to launch a thing that he calls the Center for Neurospirituality, and he's publishing papers on the intersection of religion and spirituality versus orthodoxy versus mysticism. And so, yeah, if you're interested in theology, join us too.
Awesome. That sounds super exciting. I'll have to look into that. So to wrap up with one final question, since we only have a couple minutes left. Have you each considered the ethical implications involved in creating and implementing neuroimaging indicators for brain health, and what challenges occur to you that will have to be addressed or overcome? And we can start with Loraine.
Let me think a second. Actually, I know that it's a big topic to talk about the ethical parts of especially machine learning and AI. I think there was also a question about, for example, the race. I think one of the attendees asked that. So I also had a data set where I had the race, and these are all questions. I'm not sure how to address that, because sometimes, for example, in data sets, if you come from the ethical side, people are unsure if they should use stuff like gender, race, whatever those measures are. And I don't know if it might be important for exploring health or for doing your research, but on the other side, it's also an ethical question. So yes, definitely I asked myself about that as well. But yeah, I think we also need to research in this direction, because I also saw that there's a lot of research done now for ethical machine learning, stuff like that. It's also a really interesting area. Yeah.
Great. Yeah. How about you, Sue?
Yeah, and it's an exploding field, and it's currently being developed in rapid time, and it's extremely important, and I feel very strongly about it, and things are changing in a positive way.
And you, Haoqi?
Yeah, I think our colleague Sandy, she mentioned that one of the possible issue is with the insurance company if they have access to, for example, brain age index or some others. Then the insurance company, they will know, for example, you're at high risk of some disease, and then they will make some policy change. So I think it's really important, if we, on our scientist or research point of view, we should really validate it. But on the legal point of view, there should be some, some guidelines or even some laws to enforce that who can access this information and who cannot. Yeah, thanks.
And we can wrap things up with you, Shan.
Yeah, so this is really, so I think about this stuff a lot as a clinician. There's I spend most of my time living in MATLAB, not in the clinic, and in that time, it's very easy when you're spending all your time writing code or writing algorithms to forget that the data represents real human beings. So what we're talking about here affects people in a lot of different ways, and there's a fine line, for example, between finding approaches to treat mental illness versus finding approaches for human enhancement. That's really a challenging thing. So people have tried to use a lot of these brain stimulation or brain imaging approaches for cognitive enhancement, but they could also be using it to enhance or suppress other things. Where do you draw the line between treatment and mind control? So as I mentioned, we have talked about how to enhance things like spirituality and religiosity and criminality, or attenuate criminality. What if you go the wrong way and accidentally increase criminality? These are important potential ramifications that we have to think very carefully about and avoid using these things unless we know what the outcomes are. That's why we have to do the clinical trials.
Great. Well, thank you all so much for your amazing talks in this Q&A session, and thanks to all of the attendees for coming. The next session is the state of the field, and it starts at 11:15, so you have about a 15-minute break, and then you should head over there to check out that talk.